Okay, chapter 28. When I was in college and as an undergrad, one of the courses I took was one of the more interesting courses I took was a course that was called the History of Science. And basically, the way it was just a historical account of the advancements in science. And one of the things that stuck with me, probably because the professor was saying it so many times, was that science and technology evolve parallel. That oftentimes in science we make discoveries when technology is sufficiently sophisticated enough by which is for us to make those discoveries. If you think about, the atom colliders that they have now, where we found so much about the structure of the atom. You think about what we've learned about space, simply because of telescopes and the advancement in telescopes. When I was a kid, right at the early part of the 60s, we were unaware there were other galaxies in the universe. We thought that the Milky Way, our galaxy, was the only galaxy was out there. But then with telescopes, we've learned that, as everybody knows now, we're not alone at all out there. This kind of brings me to this idea of looking at protozoans. Protozoans would have been another one of those aspects of biology that really wouldn't have been well known or discovered, if you want to say it that way, until the technology, that is the microscopes, were built well enough to be able to see those things. And you look through microscopes in Bio 1, and of course you do in this class too, and you understand that a microscope is a very precise piece of equipment. So in order to get a really good image of something microscopic that you're looking at, not only does it have to magnify the image, but it has to make it clear. We say magnification and resolution. So putting it together a bunch of lenses is not the only thing you need to do to make a sufficient microscope. You have to really take into account the clearness of the picture. And many years ago, like these two folks you see on the page, Robert Hooke. Robert Hooke is the fellow that we generally credit with the name cells. He's the fellow who gave us that word for that single entity with all those characteristics of life. And the reason why he did that is he looked at cork cells. You can see that picture on the left. That's actually his drawing. He looked at cork cells under a microscope. And like in plant cells all have these regular arrangements to them, regular sort of rectangular shapes. And to him, that looked like cells you would find in a monastery with a regular sort of shape. Now, Anton von Leeuwenhoek, he's a little more applicable to our chapter. So we're talking about, we're 17th, 18th century again. Anton von Leeuwenhoek has this really neat quote. That's his microscope down there that you see, down there in the middle. I always think it's interesting how old stuff like that, they kind of went overboard on the way they made it. They kind of made it fancier than it really needed to be. But when he looked through that microscope, the quote he had, it's very subtle, but it's just really touching to me as a biologist. He says, "No more pleasant sight has met my eye than this." I think why that touches me so deeply is I remember as a young kid, the first time looking into a microscope in a drop of pond water. I don't know, I think I was 6th grade or something like that, and just being absolutely overwhelmed at all of the life that was living in a little tiny drop of pond water. And maybe that's a little bit what von Leeuwenhoek felt about that when he looked through the microscope. Now, in spite of the fact that the discovery was made and everything's new, of course, we got to figure out what these things are under the microscope, right? It's not enough. Scientists aren't happy enough just to look at something and say, oh, this thing's swimming around in pond water. We're going to find out what they are. And at the time, of course, in terms of the kind of critters we knew existed on the planet, we knew there were animals, obviously, and we had figured out there were these things called plants, and we figured out these things called fungus. Prokaryotes are still a little bit behind, especially if you think about how small they are compared to even protozoans and the challenges with seeing them. But when we started looking into those drops of pond water and those microscopic critters, we didn't exactly know what to call them. They weren't really plants. They weren't really animals. They move around like animals, although not all of them are able to do that. And unlike animals, some of them that can move around can also photosynthesize. So what von Leeuwenhoek called them animalcules, right? And so it's a variation of the word animal, obviously in his language. I don't know, it looks like he was Dutch or something. So animalcules was the first term he gave to these little critters that were swimming around underneath the drop of pond water. Now, I've cautioned you many, many times through our evolution chapter, especially at the beginning, that just because the way something looks or the way something acts doesn't necessarily mean it's related. So von Leeuwenhoek, all credit to him, not knowing any better, he called these things animalcules because they moved around. right? Animals move around. Plants don't move around. Fungi don't move around, but animals do. So he kind of stuck with that in a very basic term. That's what he called them. And for many, many, many years, up until really recent times, to be honest with you, we lumped all of those little tiny things that swam around in a drop of pond water, we lumped them under one big giant kingdom. We called them the kingdom Protista. All right. There used to be there was the kingdom Animalia, the kingdom Plantae, the fungi, of course, the two domains in the prokaryotes, but we call them the kingdom Protista. Now, that kingdom Protista is an old kingdom name because the group Protista or protozoans are polyphyletic. If you're looking over on the right-hand side, this is some of the protozoan diversity that we're going to go through. And what I mean by them being polyphyletic, remember when we're talking about something that's polyphyletic, the group we're discussing is including the descendants, but not a most recent common ancestor. So what we mean by that is these are protozoans up here at the top. We'll talk about those, the diplomonads, the parabasids, the euglenozoa. You probably recognize that as euglena. The sarclade, SAR, is actually the stramenopiles, the alveolates, and the rhizarians, and those are all protozoans. So we got all protozoans here. The Archaeoblastida include the red algae. They include the chlorophytes and the charophytes, which are green algae. And here's where it becomes a little bit tricky. You notice mixed in this group with the protozoan are the land plants, right? So in amongst a lineage of protozoans, the red algae and the green algae, again, they're kind of halfway protozoan, sort of plant-like. You got the land plant stuck in there. And then if you jump down below to the last one, the Uniconta, that's the word I'm saying right there. We're talking about the amoebozoans and the opisthokonts. And the amoebozoans are things like slime molds and amoebas, your typical amoeba. But notice what's mixed in with those opisthokonts, fungi and animals. Now, a choanoflagellate is a protozoan. A nuclearid is a protozoan. Just like the red algae and the chlorophytes and charophytes are protozoan-like. So you see what we mean by being polyphyletic, right? Protozoans are organisms that evolved, or they're more closely related to other groups of organisms. Like the red algae are more closely related to land plants than they are to other protozoans. And the fungi and the animals are more closely related to, the choanoflagellates are more closely related to the animals than they are to other protozoans. And that's, by definition, a polyphyletic group. So a polyphyletic group is sort of an artificial designation. We call all these things protozoans, and we initially said they all go in one kingdom because they're small, and we don't know any other way to classify them, right? And that's not always a good way. Most eukaryotes are protozoans. As a matter of fact, the protozoan group is what we would basically call the stem or the base group for all the other eukaryotes. In other words, as you see in the picture there, the land plants and the fungus and the animals, the only three groups we have left, because in this course we go through the prokaryotes, which we did, protozoans today, and then we're going to move on to the plants. All of those groups, all of those other eukaryotics or organisms came out of the protozoan lineage. So oftentimes what biologists will say is the protozoan group was the base group for all other groups of eukaryotes. Protozoans, like their prokaryotic ancestors, many of them are unicellular. There are some colonial and some multicellular ones. And again, remember I cautioned about in the prokaryote chapter, when we're talking about colonial and multicellular, does not mean tissue yet, all right? So there's that sort of that gradient that occurs. Protozoans are kind of like the prokaryotes in the sense that they can make a living many different ways. You know, I showed you the metabolism of prokaryotes. They can oxidize iron, and they can oxidize sulfur, find all different things they can survive on. The protozoans are that way as well. Many protozoans are actually what we call mixotrophic. And what that means is this is an organism, mixotrophic, that can photosynthesize and engages in heterotrophic behavior. So photosynthetic and heterotrophic is mixotrophic. These organisms can reproduce asexually or sexually, and I put sexually in quotes because it's not sexually in the way we typically think about it. But remember, the technical definition of sexual reproduction is when two organisms exchange or contribute genetic information. Remember, if you're an asexual, it's you're a clone, right? So sexual means this mixing of information. Some are pathogenic, so some are disease-causing. Some are beneficial, some are mobile, some are sessile. So again, you can see the idea that lumping these all together just because they were small was a matter of convenience, and calling them in that big kingdom Protista was indeed polyphyletic, because these organisms are nowhere near. I mean, yes, among the Sarclade and the Excavata group up there, there's some relationships there. But as I mentioned, the other group of eukaryotes are shoved in there along the way. Now, prokaryotes are one of the ones that we are, when we talk about this idea of endosymbiosis, you remember endosymbiosis was one of the ways that some of the prokaryotes escaped when the oxygen levels on Earth began to rise. Remember, during the oxygen revolution, some critters couldn't survive with those higher levels of oxygen. They just don't do well with oxidation, which oxygen is very good at doing. So a lot of these critters actually entered into endosymbiotic relationships. And I introduced that to you briefly when we were looking back in some of the earlier chapters. But actually, endosymbiosis can occur more than once. Like, for example, if we think about a heterotrophic eukaryote over here that would engulf a cyanobacterium. Now, that, of course, would provide a benefit for that heterotroph because the cyanobacterium is photosynthetic. So now you got a mixotrophic critter. And then among the protozoans, secondary symbiosis would occur among the red and the green algae, so that whatever this cell that was occurred here was then engulfed again. All right, so they evolved into red and they evolved into green algae, and then those were engulfed again. And eventually we find these red algae endosymbionts in dinoflagellates, which is a group of protozoans, and stromatophiles. And the green algae are going to become the plastids, and in the chlorarachnophytes, which the arachnos, the spider, kind of looks like a spider, right? These things are going to become the chloroplasts. And if we look in some of these organisms and we look at that, what's remnant of that endosymbiotic critter that they engulfed, take a look at the picture in the lower right. This is a picture of a cell that has been engulfed, that has undergone the process of endosymbiosis. So it's got an outer membrane, which is the membrane that the cell puts around the organelle, the inner membrane, which is the actual membrane of the organism at one time, and a nucleomorph is a degenerate nucleus. And you can even see that there are some nuclear pores and gaps, all right? So that's direct evidence when we look at things like chlorarachnophytes, and we see this nucleomorph and these double membranes, that something's been engulfed inside of something. You remember back in Bio 1, all cell organelles have a membrane around them. There's the cellular membrane, and then if we're talking about a mitochondria, the mitochondria has a membrane, and then there's another membrane around it. Same thing with the chloroplast. That double membrane there, actually it's a quadruple because all membranes are phospholipids in a bilayer, right? So that double layer of a bilayer of phospholipids is indicative of something that's been engulfed, right? So this was the original cell membrane of the organism when it was by itself, and this is the membrane that was formed when it was engulfed, either through primary or even secondary endosymbiosis. All right, so very common among protozoans. Let's now turn our attention back to protozoan diversity, and let's look at this big group up at the top. Not big group, but this group at the top first, the Excavata. The Excavata are named that because they basically have a groove on the side of their body which they use to excavate, or they use to basically excavate is to, excavate, to drill into, to dig into, okay? So these flagellated organisms of this group have that groove on the side, and that's where they get their name from. Giardia intestinalis, is an example of an excavata. It's specifically a diplomonad excavata. There are three types of excavata. There are the diplomonads, the parabasalids, and then the euglenozoa. Now, interesting thing about Giardia and diplomonads is diplomonads, when you look at them underneath the microscope, are usually pretty distinctive. They have two nuclei, like eyes almost, multiple flagella, They have a degenerate mitochondria, something called a mitosome. Whenever, this is that detective part, the whole form and function thing I've been talking about, whenever we witness degenerative mitochondria, one of the things that we got to think about is what metabolic activities does this organism engage in? And you remember, with a mitochondria, you can do aerobic respiration. And without a mitochondria, or one of these degenerate mitochondria, you're pretty much limited to anaerobic. Now, many times, especially small critters like protozoans that are anaerobic, many times they're parasitic. Excuse me a second. Maybe I'm getting some pathogenic protozoan in my throat there. Anyway, many of these organisms are parasitic. For example, Giardia intestinalis is a common parasite, or a relatively common parasite among vertebrates. It gives you gastric disturbance, or upset stomach. We can get Giardia intestinalis. The interesting thing about Giardia intestinalis is we oftentimes think about things that might be pathogenic or disease-causing as living in squalid conditions, dirt, nasty conditions. But Giardia intestinalis is very commonly found in these crystal clear, open mountain streams that you might come across if you were hiking up in the woods. So it looks nice and clean, but it could easily have Giardia intestinalis. So it give you a really bad stomach ache. So diplomonads, you have two nuclei. Parabasalids have undulating membranes. They actually move in two ways. They move by undulating their membranes, kind of if you imagine like the way a stingray swims, right? These membranes move up and down, and they also have flagella. This is a very common symbiote, not pathogenic in this case. This is actually a mutualistic symbiote found in the gut of termites. Just like, the bacteria that live in the stomach of cows, just like you and I, we can't digest cellulose, but the bacteria can. So the parabasalids, these organisms living in the gut of termites, do the same thing as the bacteria do living in the guts of cows. They allow the termites to digest cellulose. Cellulose, a major component of wood, right? And again, a degenerate mitochondria. In this case, we call it a hydrogenosome. That's just another word for a mitochondria that has lost its overall function of aerobic respiration. So again, we're going to be assuming that these guys are anaerobic, right? Now, there's a parabasalid that is particularly problematic to human beings, and we'll look at it in lab. It's one called Trichomonas vaginalis. And Trichomonas vaginalis is a sexually transmitted disease. It's an STD. It's a silent STD. This is like chlamydia, when I was talking about in the prokaryotes. What does it mean when an STD is silent? What does it mean when a disease in general is a silent disease? What do we mean by that? Or what does that sentence mean that sometimes it's silent in males? Yeah, asymptomatic, so it means you can spread it, right? Asymptomatic, so when it's silent, it can be spread. So Trichomonas vaginalis, typically a protozoan infection that would affect females, hence if you look at the name, it also can occur in males. But being silent, the problem is males don't know they have it, and they can continue to spread. As a matter of fact, this is the most prevalent parasite in North America, with nearly 5 million cases a year. It's not difficult to treat. It's not deadly, but it is something that is spread through sexually transmitted, through sexual contact. All right, and the last one, the Euglenozoas in the excavata group. Those are the Euglena. These were some of the earliest eukaryotes that actually had mitochondria. This is a mixotrophic organism. Not all euglenas are mixotrophic, but about 30% have this ability to photosynthesize. Reproduced by mitosis. There are free-living versions. They have in their flagella, they have a crystalline rod structure. Now, all flagella, you remember from Bio 1, are made of these triplets of microtubules. So nothing unusual there. But this crystalline rod that they find in their flagella, function unknown. Not sure exactly what it does. I find that interesting too, right? Because we think that sometimes in science everything has been discovered, and we find out that that really isn't necessarily the case, right? In this particular situation, that crystalline rod must do something, but we're not really sure what kind. Okay, now another type of euglenozoa that I want to talk about is belongs in a group called the kinetoblastids. All right, so there's a typical euglena that we'll look at in just a second, but I do want to mention the kinetoblastids. The kinetoblastids have a single mitochondria. It's called a kinetoblast. Many of them are parasitic. For example, one of the genera of kinetoblastids that's well known for being parasitic are the trypanosomes. The trypanosomes causes Chagas disease, or there are other trypanosomes that cause African sleeping sickness, which is spread by the tsetse fly. This is the tsetse fly. It's the vector. So again, it's a blood sucking organism. So when it begins to take a blood meal from you, the first thing that these organisms do that feed on blood meals is they inject a little bit of an anticoagulant into the host, so the blood flows nice and freely. Well, when they inject that little bit of anticoagulant, very often these little protozoan trypanosomes, these little purple things, will be injected into your bloodstream as well. So you see, those are the red blood cells. This is a blood parasite. African sleeping sickness is a problem in human populations. The reason why it's called African sleeping sickness is because many times the transmit between the flies is between humans and the cows, the cattle that they have in their livestock. And since these people very oftentimes live very close to their livestock, then the transmission of disease is much higher. The problem with this trypanosome in your blood system is, of course, your body sees it as foreign, so it's going to try to get rid of it, right? Well, the only way your immune system is going to know whether something's foreign or whether it's supposed to be there or not is by sensing the receptors on the outside of the cell, right? The glycolipids and the glycoproteins. You might remember that from Bio 1. What the trypanosome does is it tries to stay one step ahead of your immune system by constantly changing its protective coating. So like in the prokaryotes where you can hide your protective coating, you can also change your protective coating so that you basically are tricking the immune system into saying, oh, no, I'm not a trypanosome, right? Until the immune system figures it out, begins to attack them, and then, again, the trypanosomes change their protective coating. Very common thing that happens in parasites, to hide from your immune system. And if a doctor suspected of you of having African sleeping sickness, what he would do is take a blood sample, put it under a microscope, and he'd look for these little purple squiggly things, which I'll be able to show you in lab too. But most of euglenozoas are the euglenids, right? The typical euglenids. Euglenids have two anterior but unequal flagella. They have a short one and then a very long one. The long one is the one we typically see. Remember, in organisms that move around with flagella or cilia, flagella is usually one or two or maybe a half dozen, and they're bigger, right, longer, whereas cilia are generally much shorter. You generally have lots of them. So this is how this organism obviously moves around. Contractile vacuoles for osmoregulation. So this tells us right away that euglena are found in freshwater environments, hypotonic environments. They have this thing called a pellicle. Where is it labeled? Oh, there it is. The pellicle is a band of protein that wraps, you can see it, it's wrapping around the euglena, and it gives it strength and allows it to be very flexible and move around. This is another example of that secondary endosymbiosis. The chloroplasts that are found in euglena, which allows them to be mixotrophic, came about because these organisms ingested green algae. So remember, what we had is an organism that ingested some sort of protozoan like a cyanobacterium. Then that was ingested by a green algae, right? And then the green algae was ingested. Secondary endosymbiosis. Now, before we move on for just a second, why do you think there's an eye spot or light detectors in these organisms? Why would these organisms need an eye spot or a light detector? Think of the eye spot as being really a very simple light detector. Why would that be something you would expect to find in this critter? A little detective work, right? It's critter makes a living how? In other words, how does it get its nutrition? I said it was mixotrophic, so it's heterotrophic, but it's also what? Absolutely. So it's autotrophic, okay? So if you were autotrophic, photoautotrophic, you would have to know where the photo is. You'd have to know where the light is. So remember when we were talking about exaptations? This is not an eye that Euglena could read the paper with, but it certainly is an eye that works great as a light detector. And if you're a photosynthetic critter, that would be something very important to know. Where's the light? Where's most of the light? Especially if you're living in a lake, you want to know when to swim to the top of the lake, right? Okay, so that pretty much covers that first group, the excavata. I'm going to move now to the SAR clade. The SAR is an acronym, again. The Stramentopiles, the alveolates, and the rhizarians are the three members in the SAR clade. And we got diatoms, golden and brown algae. Let me say once again, be careful with that word algae. The word algae can refer to many different things. There were blue-green algae, which is a prokaryote. Here you see there are golden algae, brown algae, red algae. Those are protozoans. Green algae, in which some of them are protozoans and some of them are plants. So algae can be a prokaryote, could be a protozoan, or it could be an actual plant. So, don't get confused when you see that word. That word doesn't necessarily mean there's any indication of relationship between them. So among the Stramentopiles, we're going to look at the diatoms and then golden algae, brown algae, and the alveolates contain the dinoflagellates, the apicocomplexans, very interesting group. That's another parasite group. The ciliates, right? The paramecium, the ones that the very common protozoans. The forams, the cercozoans, and the radiolarans are in this group. So let's get into it and take a look at the Stramentopiles first. If you're a member of the Stramentopila group, the word Stramentopila literally means hairy flagella. Okay, so this is a Stramentopile. And you can see that Stramentopiles very often will have two flagella. One has hair on the end of it, and one is smooth. This is what it looks like a little bit closer. These are little hairs. This would just add more resistance as this organism moves through the environment. There are some Stramentopiles out there that have nothing but smooth flagella. But again, it goes back to that thing we talked about early in the semester about looks being deceiving. When we look back at the genetics, when we look back at the phylogeny, those Stramentopiles with smooth flagella used to have hairy flagella, so they lost them. So that's always something you got to be careful with. If an organism has a particular trait, or if an organism is very different than everybody else in that group, it could be like we were looking, with photosynthesis in that. Did photosynthesis arise first and then get lost later on, or was it the other way around? So characteristics can be lost. Diatoms are Stramentopiles. Diatoms are some of the neatest critters out there. They're all kinds of different shapes. Over 100,000 species, photosynthetic, unicellular. They have these shells that are made of silicon dioxide, a major component in sand. And they fit together kind of like a shoebox. So this is the top and the bottom. That's how these two valves would fit together. The shell is made of silicon dioxide because it allows these organisms to stand up against pressure. Many of these organisms are found in the deep ocean. Many of them can withstand pressures up to something in the vicinity of 33 million pounds per square feet. It's amazing to think about that pressure. Over on the right, if you wonder what the styrofoam cups are, just to give you an idea of the pressures that we find at near the bottom of the ocean, that's a normal size cup on the left, and styrofoam is mostly air. And the one on the right is one that was brought down in a deep submersible and exposed to the pressure of the water down there, and you can see what it's done. It's just kind of shrunk it all down, just basically squeezed all the air out of it. Diatoms, especially the shells, can be ground up to make something called diatomaceous earth. If you're an aquarium person, you may have heard of diatomaceous earth. Diatomaceous earth is a very good filtrant. It's very good at filtering impurities. There's a lot of things for things to stick to. these organisms too, these diatoms, are a major source of where carbon occurs during a bloom. So when these things bloom and their populations start growing incredibly, that's where a lot of the carbon goes, which can be a problem if this is happening, say, in a freshwater environment. As the diatoms bloom and start growing, and we talked about this in the prokaryotes with algae blooms and like Apopka. Not only do they remove the oxygen, because they need oxygen too, but they're also removing a lot of the carbon, because as these things are blooming, they're growing, and carbon is the building block. So removing the carbon from the environment makes it difficult for other critters that need carbon. All kinds of different shapes of diatoms. Very neat. Golden algae are photosynthetic. They get their name not from green chlorophyll, but actually from carotenoids. Carotenoid, you remember, is another photosynthetic pigment found in carrots, so sort of an orangish color. Golden algae have two flagella, although they all use these carotenoids for photosynthesis. Some of them are mixotrophic. Some are unicellular. Some, like Dinobrion there on the right, are colonial. Colonial means you live together. You're not multicellular, though, of course. And like a lot of other critters that we've talked about so far, and critters that we will talk about as we continue, when the environment changes, right, you can go extinct, you can adapt, or you can just wait it out, right? Kind of like we were talking about the anthrax. You form a spore, and you wait it out until the environment becomes a little more conducive. And these spores or these cysts can last various lengths of time. Now, again, eventually, if the environment doesn't get conducive, these cysts and spores will deteriorate. But in some cases, these things can last decades. All right, so there's the golden algae. We got the brown algae. Click. There we go. There we go. Brown algae are the kelps. We call them seaweeds, but they're not plants. Even those huge forests of kelp that you see out in California, these can be, in some cases, these can be 30, 40, 50, 60 feet long, these long expanses of kelp. They are attached to the bottom. I'll come back to this slide, but they're attached to the bottom by something called a holdfast. And then what holds them up is a stipe. And then the things at the top are called blades. Now, I know this looks a lot like a plant, so you might be inclined to think roots, stem, and leaves. All right, but look over here. I asked the question, do these things look like plants because of common ancestry with plants, or because of convergent evolution, or are they analogous or homologous structures? Now, these are photosynthesizers, and they have to hold on to something, and they have to grow up towards the light. So the stipe can grow longer so they can get up towards the light. They also have these nematocysts right here, which are air floats. These things can float. Right, so these are analogous structures with plants, right? So to be technically correct, we don't use the word roots, stems, and leaves, because that would be for plants, and this is a protozoan. So we call it a holdfast, a stipe, and a blade. Okay. Kelp beds are really important nurseries too. A lot of fish, the young will develop in kelp beds because they can become very thick, and so there's a lot of hiding spots. When I lived out in California, when I was in the Navy, we used to go diving in the kelp beds. It was really interesting because, like a nursery, there's all kinds of critters everywhere. And I think this is a neat shot. This is a beluga waiting outside of a kelp bed. Yes, they can. Yes, they can. Now, that beluga is waiting for one of those little fish to get a little too far from the kelp bed before he gets his meal. I know I told this story in Marine Science Ace, but I don't know if I told it, obviously everybody else. Diving in the kelp beds, sharks, one of their attack strategies, you will, is catching fish off guard. So a shark, mako sharks, for example, which are basically smaller versions of white sharks, will dash in through a kelp bed really quickly, trying to surprise some fish and getting a meal. So from a diver point of view, you'll be sitting in these kelp beds, and all of a sudden, because they're very thick, they're like a forest, this mako shark will go whipping by you real quick, while it's dashed in to try to find something to eat. So That's kind of interesting, a little bit unnerving, but kind of interesting. And yeah, Ace is right. You can actually hide in them, even if you're a good size, which would be very effective if a shark was chasing you. Okay, so again, back to my question. Are these structures that we see homologous or analogous? These structures that we see in this plant-like looking thing. Now, remember, the deciding factor, homologous would mean that they look alike because of common ancestry, common recent ancestry, and analogous would go hand in hand with convergent evolution. They look alike because they have a similar environment. Thank you. Definitely analogous. Definitely. Give me another example of an analogous structure in another critter. Someone else. So much you remember from our early days here. Kaylee, or Daniela, or Ace. Give me another example of an analogous structure. The fins, right? Okay. The critter itself wouldn't be the analogous structure, but the fins. Right. Exactly. Wings of a bat, wings of an insect, right? Same idea. Both have wings, but they're not closely related to one another. All right, now, I need to talk about reproduction in organisms, and this type of reproduction is something that's going to stay with us for a while. The type of reproduction I need to discuss is a form of reproduction called alternation of generation. All right. So let me show you what I mean by that. In a very simple, basic form, alternation of generation means that there are two multicellular stages in an organism's life. Two multicellular stages in an organism's life. Now, if you're trying to think, what does he mean by that? Think about yourself. You have one multicellular stage, and then you have one single-celled stage. The multicellular stage is when you're in the diploid form as the adult. During meiosis, you produce haploid single-celled entities, right? Sperm and egg are single-celled. What we mean by alternation of generation is organisms that do it this way have multicellular diploid and haploid generations. Take a look at here. This is laminaria. This is laminaria is a type of a brown algae, a kelp. Laminaria, you'll see it in the lab. It's slick. It always reminds me of laminate. That's how I remember the name of it. Laminaria has, we'll start with the more familiar type first. It has a diploid sporophyte. See, diploid is in the orange, so this is a 2N. This is called a sporophyte. And sporophytes, the word phyte means a structure with something on it. So sporophytes are structures that make spores. On sporophytes, there are sporangia. And sporangia produce spores. Spores are resistant structures, and it produces those spores through meiosis. So what we get here is that genetic variability. Now, the type of spores that are produced have flagella, and spores that have flagella that can move, we give the name zoospores. It doesn't necessarily mean it has anything to do with an animal. This is not an animal here, but again, the movement aspect of it, right? The same reason von Leeuwenhoek called them animalcules, because they moved around. So zoospores that can swim that are flagellated, or spores that can swim or flagellate, they're called zoospores. Oh yeah, except those spores on the mushrooms, Kaylee, those are not flagellated. So they would just be regular spores. They're the wind disperses them. Yeah, typically you're only going to have a flagellated zoospore if you live in a liquid environment, which laminaria seaweed does, right? Okay, now the difference, of course, is on the other side is what we call the gametophytes. Now, sporophytes produce spores, and gametophytes produce gametes, okay? But the difference is, in the alternation of generation, the gametophytes, the one there in the red, are haploid but multicellular. In the alternation of generation, we have two multicellular stages. The diploid sporophyte is multicellular. The haploid gametophyte is multicellular. Now, we're going to continue with this trend as we make our way through plants. In essence, the benefit of having a multicellular gametophyte, there are a few benefits, but one of the biggest benefits is you have lots of cells producing gametes. Think about a multicellular gametophyte as being a gamete producing factory, producing tons of sperm or tons of eggs. The idea again is being numbers, right? So in this alternation of generation, we're really focusing on more making lots of gametes, lots of egg, and lots of sperm, because for reasons that we've talked about a million times already, most of that stuff is going to be destroyed, right? Most of those sperm and egg will never find each other. And even if they do, many of them, even if fertilization does occur, many of them will never live long enough to reproduce themselves. Remember that Darwin said that. More offspring are created that can possibly hope to survive. So the way you can work within that system is make lots and lots and lots of gametes to get lots of offspring. If indeed a sperm and egg do get together, then we're back to the typical zygote, right? Now notice the zygote, the diploid is growing right out of the haploid, the gametophyte, and eventually that zygote is developed into a developing sporophyte, and it becomes finally a complete sporophyte. If the generations look alike, in this case they don't look alike, we say that they are heteromorphic. And if the generations look very similar, the haploid and the diploid, it's called isomorphic. So laminaria here would be an example of heteromorphic. Right? This is a heteromorphic alternation of generations. Now again, before we get off this, let's contrast that with animals. Animals have a multicellular diploid stage, but we have a unicellular haploid stage. We don't produce gametophytes. We don't make sporophytes either. We don't make spores, right? Animals don't do that. Okay, so where do we, oh, the alveolates next. So we've gone our way through the stramenopiles. Now let's take a look at the alveolates. Oh, this is kind of cool. Now, alveolates are called that because they have alveoli, and if you've taken A&P or any of those classes, you're talking about human anatomy, alveoli are the openings in your lungs, little sacs where air collects and gas exchange can occur. In this case, these alveoli don't have anything to do with respiration, but they're these vesicles that are found up near the end of these organisms. They might function similar to Golgi bodies, we think. We're not exactly sure what they do. But if you remember what Golgi bodies do, Golgi bodies secrete materials. They secrete substances. And I want you to kind of sort of keep that in mind, because that's going to become important a little bit later on when we talk about a particular type of an alveolate. Okay, now the alveolates that we're going to discuss, one of them is known as dinoflagellates. Dinoflagellates have two flagella. One flagella goes this way, and one flagella goes that way. And when they're moving their flagella around, they're basically spinning. That's where they get their name. Dino means to whirl, so dinoflagellates spin when they move around. Most of these are photosynthetic, unicellular, shells of cellulose. Ah, that's interesting. Right? Diatoms had shells of silicon dioxide. Dinoflagellates have shells of cellulose. Now, cellulose is not as strong as silicon dioxide, but it still gives you some structure, right? Interesting thing about that, this is another example of convergent evolution, isn't it? Because plants use cellulose, don't they? They use cellulose for the same reason that the dinoflagellates do. Now, these organisms are generally found in water or aquatic environments. Planktonic. Think about this word for a second. Lots of things are called plankton. By definition, something that is a plankton is a floater. Anything that floats is a plankton. So plankton can be bacteria. Plankton could be protozoans. Plankton could be small little animals. They could be larval forms of animals, for example. But if you're a floater, it's considered being planktonic. If you're a swimmer, then, I got two of you from Marine Science Center, don't I? Ace and Daniela. When you're a swimmer, what's it called? What's the opposite of planktonic? So a fish would not be planktonic. Fish would be, yeah, beautiful, nectonic. Nectonic. Nectonic means you can move around. Planktonic means, now, Planktonic organisms like these dinoflagellates, they can use their flagella to move a little bit, but we're talking about, large changes in position, large changes in where you are, and you would have to have fins or flippers to be able to do something like that. Some dinoflagellates are luminescent, and of course, living in Florida, these are the ones that cause the red tide. This is the red tide dinoflagellate. It is photosynthetic. Red can be used to absorb sunlight, not as commonly as green, but if you think about, we mentioned a second ago, red algae photosynthesize. They're not green, they're red. And when these things find themselves in a conducive environment, they bloom, right? They start reproducing like crazy, and they reproduce so much, and there's so many of them, they actually start to turn the water red. It's what we call red tide, isn't it? And what happens with that is the toxin that's produced by these chemicals the toxin that's produced by these dinoflagellates begins to become concentrated in that area, and it's deadly to fish. It can kill fish. And if you've ever been to the beach here in Florida during or after a red tide event, you walk down the beach, there are lots of dead fish, aren't there? Lots of dead sea creatures. Humans get affected by it too. Especially, I've been at the beach before when there's been a red tide event and then it's windy, where there's a lot of ocean spray. So what we're basically doing is that toxin that's in the water is being made into an aerosol. Next time you're at the beach, if it's a little bit windy and there's a red tide going on, walk up and down the beach and notice how many people are coughing. It's a respiratory irritant. The coughing that people are doing is because of the toxin. Now, you would have to actually be in the water to absorb enough to make you sick, but you do get it into your lungs. Yeah, during heavy red tide events, you definitely want to stay out of the water. I don't know exactly what the lethal toxicity, it's kind of off topic, but you guys know how they test materials to see if they're lethal to living things. What they do is they conduct what's called an LD50 test. An LD50 is lethal dose 50. What that means is how much of a substance does it take to kill off half your subjects. So if you've got 100 rats you're testing, how many milligrams of this material would it take to kill off 50% of them? It's just a way we measure and standardize toxicity. I don't know exactly what the LD50 is for dinoflagellates when it comes to humans, but nonetheless, you would have to get, I would imagine, a pretty good amount of it. One of the big problems is those who eat raw oysters. I love raw oysters. Organisms like that are filter feeders, if they live in an area where there's a red tide event, they will actually accumulate the toxin in their tissues. So you come along and you eat that raw oyster, and you now have the toxins inside of you. Again, I don't know of any deaths from humans through red tide. There may have been, maybe someone with a compromised immune system or something like that. Okay, now, still sticking in with this group, we got the apical complexes. So we're still in the alveolates, okay? And the alveolates, as I said, have these little flattened sacs up near the end. This particular group of organisms, the apical complexes, are animal parasites. So I want you to look down here where these alveoli would be. There's the apex. It's kind of like a peak. Notice what it's attached to, a red blood cell. Right? So this is a parasite. A merozoite is one of these developmental stages among these organisms. Okay, so that apex is an apical complex. It has this unique arrangement of organelles. Again, maybe kind of works like the alveoli, where it's a Golgi apparatus, right? Where the Golgi apparatus secretes material, and what it secretes are things that allows it to invade the host cell. This is how it gets into the host cell. So that merozoite that's attached to that red blood cell down there at the apex, those alveolates, those little flattened sacs, may be producing materials that are then exported out, secreted out, that are then digesting through the membrane of the red blood cell. It's a parasite. The organism Plasmodium. Plasmodium is the organism that causes malaria, and that's a protozoan. A merozoite is one of the stages in the Plasmodium parasite. Parasites many times have very complex life cycles. I studied one in South Carolina one time that was in the fish I was working on, that basically the way it worked is the parasite in the fish would lay eggs.
the fish would then, through fecal, would drop those eggs. Those eggs would hatch into a larval form that would infest snails. It then, inside of the snail, would develop into the next larval form. It would then be shed out of the snails, and then it was in. No, wait. Then it was into the fish. So whatever that came out of the snail would then infest the fish. I spoke wrong about the fish depositing the eggs of the parasite. I've got one more step. The fish then has the parasite. Waterfowl, ducks come along and eat the fish. Now they have the parasite, and it changes again, right? It modifies itself again. And then the eggs coming out of the duck would be how the parasite reproduces itself. Very complex life cycle. What we call intermediate hosts. The duck was the final host, and the fish and the snail were the intermediate hosts. So this is what we mean by a complex life cycle: sexual reproduction and asexual reproduction, many different hosts that are required. To get rid of these kinds of things, one of the easiest things to do, or one of the more common things to do, is get rid of the vector, right? Kill the mosquitoes. That should be easy enough. Now, I've lived in Florida 30 years, and they've been spraying for mosquitoes for 30 years, and it seems like there's more now than there's ever been. So getting rid of mosquitoes might be a difficulty. If you use DDT, which you're not supposed to use DDT in this country, obviously, but it's still used in other parts of the world. If you use DDT, then you basically could potentially be making it worse, right? Because when you use DDT, what you're doing is you're artificial selection, right? You're prompting organisms to adapt and change. Drug development and vaccines are one of the ways that we can help with this malaria parasite. Let's take a look at how what the life cycle of this Plasmodium malarial parasite is like. Okay, up in the upper left is the mosquito. So the mosquito draws a blood meal, and of course, being a blood feeder, it has to inject a little bit of that anticoagulant in you first. Okay, all right. So let's start off inside the mosquito with the sporozoites. All right, so inside the mosquito, meiosis occurs. Now, whenever you see meiosis, always think variability. That's the reason behind meiosis. If you don't need variability, just do mitosis. So this is variability, right? So we get a bunch of variable sporozoites coming out of an oocyst. This is inside the mosquito. Mosquito lands on you, injects a little bit of that anticoagulant. Guess what's in that anticoagulant? Sporozoites. Just like the trypanosomes, you know, the tsetse fly, same idea. So a little bit of those sporozoites enter in your body where they migrate their way to the liver. In the liver, they transform and become merozoites. That was the picture we saw at the beginning. And the merozoites attack red blood cells. And what they do is they invade into red blood cells, where they then create inside the red blood cells more merozoites. And every once in a while, the red blood cells lyse. And these merozoites that come out of the red blood cells, it's like a chain reaction, then are capable of infecting other red blood cells. Folks who have malaria, one of the symptoms of malaria when they're having a malaria attack is alternating fevers and chills. Remember Alfred Russel Wallace, the guy who, with Darwin's theory, he was in a hallucination from heavy fevers from malaria, from a high fever. That's where he came up with the theory. Remember, we talked about that. So when the red blood cells are lysing, that's when you're getting the chills, and then the fever occurs. Now, not all those merozoites are merozoites. Some of these are gametocytes, and this is how the cycle is complete. Gametocytes eventually evolve into gametes, and the female gamete looks pretty much like an egg. The male gamete is actually little squiggly things here. One cell will produce many male gametes. Now, this is going to happen inside the mosquito. There it is again. So mosquito lands on you, takes out a blood meal, and in this case, because you have malaria, you have the merozoites, or the gametocytes specifically, now enter into the mosquito. They then make their way after fertilization to the gut of the mosquito, and that's where they develop. So fertilization forms the zygote, and all this is happening in the gut, and of course, then the oocyst ruptures with the sporozoites, and the cycle's complete. So you see, we got two hosts here. We got the mosquito, we got the human, and we have the sporozoites, the merozoites, and the gametocytes. We have three different types of infectious cells. Right? The sporozoites infect the liver infects the merozoites infect the red blood cells, and the gametocytes are then used to complete the process by reinfecting the mosquito. Malaria, not such a big problem in this country. Big problem in other parts of the world, though. Especially parts of the world in which mosquito eradication isn't as far along as it is in other areas. A lot of standing water. It's like the same thing with the Zika virus. People with standing water, mosquitoes love standing water. So get rid of the vector, you get rid of the problem. All right, the ciliates. The ciliates are the cilia, paramecium, which always to me looks like the bottom of a shoe. Paramecium are cilia, or they are ciliates. Long rows of cilia all around the outside, not just on the edge, folks. It's all around the outside of this organism, used for movement back and forth. Very, very agile. Cilia are very well choreographed, and these organisms can move back and very flexible. Two type of nuclei, though, in ciliates. That's unusual. They have a macronucleus and a micronucleus, a big one and a little one. We'll talk about those in just a moment. Food vacuoles, obviously. The oral groove opens up into the mouth of the cell or the opening. Contractile vacuoles, they live in fresh water. So you always have to worry about water pouring in. There's another contractile vacuole. So water pours in a freshwater hypotonic environment, and the contractile vacuoles squeeze out the excess water. Now, this whole thing about the micro and macronucleus actually has to do with generating variability in these organisms. Asexual reproduction is fine, right? But there's no variability there. A little bit of variability. Every time the DNA makes a copy, remember, it's not exactly the same necessarily. But you don't get as much variability out of asexual as sexual. And I've hammered since the beginning of the semester how important variability is, right? You got to have variability. You got to have differences so that there is something for evolution to work with, for natural selection to work with. You got to have variability because you got to have some that are fit and some that aren't so fit, right? So let's take a look at how these ciliates, very often improve their genetic variability, or they add to it. All right, paramecium ciliates can engage in something called conjugation. Conjugation is just basically they're getting together. Okay, see in the upper right, these guys are in conjugation. But let's start with number one, what's going on. All right, first of all, we say two paramecium of different mating types. Paramecium don't have sex. There's not a male and a female. So we just basically call them mating types. So we're going to have the purple mating type and the red mating type. Okay, they get together and they decide they're going to conjugate, however they decide that. And meiosis then occurs, variability. Now, meiosis doesn't occur to the big nucleus. It just occurs to the little nucleus. So the little micronucleus divides by meiosis. And of course, when you divide by meiosis, you end up with 4 cells, right? One cell divides, then divides again, so you get 4 cells. So you got 3 haploid micronuclei. Now, of those 3 haploid micronuclei, only one of them remains. The other 3 disintegrate. Now, why were they even formed? Well, you're shuffling up the genetic information. It's going to be random which one of those micronuclei sticks around and which one of the 3 disappears. Okay, so it's all that variability thing. Now, the next thing that happens is in each cell, in each paramecium, the micronuclei divide. Now, the reason why they divide is when conjugation finally occurs, what they're going to do is they're going to swap micronuclei, right? Mates exchange micronuclei in conjugation. See right here? We've swapped those micronuclei. Now, once that happened, the micronucleus, okay, fuses with the micronucleus that's already there and forms a diploid micronucleus, all right? So this is haploid. This is haploid. They're going to fuse and form a diploid micronucleus. That diploid micronucleus is going to undergo mitosis. The macronucleus, in the meantime, disintegrates. And the 2 micronuclei, one becomes the macro and one becomes the micronuclei. All in all, what is happening here, even though it seems like a relatively long, convoluted way of doing it, is we're shuffling up genetic information. We're doing it here when we do meiosis. We do it here when randomly 3 of the micronuclei disappear. And of course, there's the randomness of which 2 paramecium get together as well. So it's very similar like in animals, right? Sperm and egg are different each time in meiosis, and human beings, there's variability amongst them as well. All right, so it's not really sexual reproduction. That's why I put it in quotes. It's definitely not asexual, but it is sexual reproduction in theme. Maybe that's the way I should say it. Sexual reproduction in theme. What I mean by that is sexual reproduction is genetic information being combined from more than one individual, and that's what you're doing here. You're just kind of doing a little bit different way. You're swapping the genetic information. Let me see where we are. Sarclade. Let's do the Rhizarians. Yeah, we'll do the Sarclade, and then we'll finish where we 305. Perfect. Let's just finish up with the Rhizarians, the last group in the Sarclade, and then we'll move on. Yeah, that'll be good. Make sure we don't lose our time. All right, the Rhizarians. Many species of Rhizarians are amoebas, but the word amoeba is not monophyletic. Okay, so lots of things are called amoebas. It doesn't mean they're related to one another. So amoeba would be another example. If it's not monophyletic, this would be another example of a polyphyletic group, right? Polyphyletic group. This is an amoeba, that's an amoeba. They all look kind of like the same, but they're not necessarily related to one another. Rhizarians have these small little pseudopodia. Remember the amoeba, like in Bio 1 that you learned, amoebas have blunt pseudopodia, right? This is an amoeba, and this is a pseudopod. These have very long, thin, thread-like pseudopodia, which they use for food capture. Many of these live in the water, aquatic, right? So those thread-like pseudopodia help spread out the surface area. It helps them float. It's kind of the same thing like if you're floating in water, you put your arms out and your legs out, and you're trying to increase your surface area. It gives you a little better flotation when you do that. Now, amongst the Rhizarians, Radiolarians fit in this group as well. Radiolarians are very neat. They're marine. That means they're freshwater with a skeleton of silica, which is glass. This right here is the shell, okay, which is made of silica. The shell is called a test in this context. And these pseudopods are these little things that are sticking through the shell. So here's the shell again in this one, and all these little pseudopods are sticking through. Great for capturing food, great for defense, right? All these little things sticking out. Look at all these little needles sticking. Hello, I'm back again. Sorry about that. I thought I was going to make it through the whole class without that happening. Okay, so Radiolarian, silica skeleton, silica test, a lot of little pseudopods sticking out all through a bunch of little holes that you find in that test. The foraminiferans, the forams, these are heterotrophic. Here's what they look like. Again, we have a test. In this case, the test is made of calcium carbonate. Calcium carbonate is a very common thing to make shells out of in marine organisms. A lot of calcium in the ocean, a lot of opportunities, like the shells on clams and oysters, calcium carbonate, same stuff. Very thin podia that they use for swimming and feeding. They have an alternation of generation, so they have a life cycle with haploid and diploid generations. This is the White Cliffs of Dover on the southeastern coast of England, just across the channel from France. Well, maybe not just across, what, 25, 27 miles, something like that. And the White Cliffs of Dover are basically remnants of millions and billions and trillions of foraminiferans that have died and formed these layers, and then through geological activity or tectonic activity, this part of the rock layer was pushed up above the water. These organisms live in the water, so they died, they decomposed, and all of the white you see there is the remnants of the calcium carbonate shells. Now, there was something that used to be in classrooms many years ago. They don't use it anymore, but there was something in classroom that we used, teachers used, that was directly connected to foraminiferans. Does anybody know what it is that was used historically in classrooms long before there were whiteboards? Look at the White Cliffs of Dover. Does that remind you of anything you may have seen in a class? Yep, not the chalkboard, Ace, but the chalk. Yeah, that's what chalk is. Chalk is the calcium carbonate tests or skeletons of these foraminiferans. Isn't that neat? Okay, last group, the cercozoans. This is a very morphologically diverse group, all kinds of different type, very common in soil, pretty much found everywhere. Marine, freshwater. Let's talk about that word marine and aquatic for a second. Marine means saltwater. Okay, so an organism that lives in a marine environment is living in saltwater. Aquatic is a little bit vaguer in the definition, but many times aquatic refers to organisms that are freshwater. So there are aquatic fish and marine fish, okay? Cercozoans are soil, live in terrestrial. They're marine. They're also aquatic, so they live in freshwater. Move around through flagella or pseudopods. These are a great example of endosymbiosis. This is Polynella chromatophora. This is a protozoan, folks. Okay, so this is a protozoan. And inside Polynella chromatophora, there's a chromatophore. Chromatophore is green, helps it photosynthesize. So this is like a chloroplast type thing. But the reason why we have evidence to suggest that chromatophore came about through endosymbiosis is because when we look at the chromatophore, it's surrounded by a peptidoglycan membrane. Okay, folks. Now, where else, what other type of critters have peptidoglycan membranes? Of course, this is presuming that you either listened to the chapter 27 lecture or you had a chance to get into chapter 27. Does anybody recall that word, peptidoglycan? Where do we typically find those kinds of membranes? Not where do we typically, where do we only find those kinds of membranes? No. Domain bacteria, folks. Peptidoglycan is a unique structure only found in members of the domain bacteria. So if we're looking at that chromatophore and we see that peptidoglycan membrane doing a little bit of that detective work, we're going to go, I'm guessing this is endosymbiosis. So that chromatophore was a free-living bacterium at one point, many millions of years ago, and some protozoan critter engulfed it through endosymbiosis, and again, for reasons that we've talked about, that was a benefit, right? You can photosynthesize now. That's definitely a benefit. Okay, I'm going to stop there for now. Let's see, we need to, there we go. We'll move on. We're done with the sarclade. So when we get back together, we'll talk about the archaeoblastids. We won't talk about the land plants because we're going to talk about those when we get into the plants. And then we'll move down here to the Uniconta group. The slime molds. Very cool. Very weird critters, these slime molds. But I'm going to stop there for now. Remember, again, you do have your exam on Monday. It will start at 2 o'clock. It will be here on campus, and it'll be done in the computer lab upstairs. Chapters 22 through 26. Make sure your assignments are in by 8 o'clock that morning, too, okay? All right, folks, that's all I have for now. Let me stop the recording. If you have any questions, by all means, stick around. If not, have a good weekend, and I will see you guys on Monday. We are doing lab number 4, so we're doing the protozoan lab on Monday.